Over to the cloud. Okay, share screen. There we go. Boom. Let's make sure this works out right. Awesome. Okay, good. Okay. Welcome to hell. Yeah, we are now firmly entrenched in the craziness of metabolism. So the first 20 or so slides out of the 104 that we have here are somewhat background and less necessary to what I actually want you to know, but we're going to fly through those slides and then pause a little bit and slow down for some of the next ones. Okay. So in the previous chapter, we talked about the biosynthesis of lipids and fatty acids in particular. So now we're going to talk about the synthesis of amino acids and nucleotides. And of course, anything that's related to those, because you can often use those as precursors to make other biologically active molecules. So before we get to that, we're going to start talking about nitrogen. So just like with the urea cycle, where we talked about eliminating nitrogen from the body, here we are using nitrogen to incorporate it into molecules. Okay. The trouble is that how do we actually get nitrogen in there? Okay, so we found our nitrogen. It's one of our was it chomps or ponchos? That's what I like for phosphorus, oxygen, nitrogen, carbon, hydrogen, and sulfur are probably our 6 primary elements in organic and living organisms. So nitrogen is one of them. It's found in a ton of stuff besides our nucleic acids and our proteins, cofactors, hormones, neurotransmitters, pigments, defense mechanisms for mushrooms, like a manatin in there, our angel of death mushroom in here. But how do we actually get it in there? If we look at molecular nitrogen, our atmosphere is 9% nitrogen. We should be able to incorporate it everywhere, right? But N2 is pretty much inert. There's not an easy way to get into any compound. Haha, it's a joke. Oh, terrible, right? Okay, so what we really need to do is to combine our nitrogen with hydrogen to make ammonia, and ammonia is much more reactive, okay? And this should be a very favorable reaction to combine nitrogen with hydrogen. It's got a large negative delta G, but because nitrogen is a triple bond, it has a massive activation energy. So if you think that reaction curve, you have this huge speed hump there. So, if this were an easy reaction, our atmosphere would just explode into ammonia. And that'd be kind of cool, but we'd all be dead. So there are, other processes that can cause this to occur. We can look at combining nitrogen with oxygen to make nitrous oxide, and that happens with lightning. You can also get ozone with lightning as well too, but that's a massive release of energy there that can overcome that activation energy. Civilization really kind of took off in around sometime in the 1800s, I think, with the Haber process, where we figured out how to combine nitrogen gas with hydrogen gas to make ammonia. And this allowed the creation of various fertilizers, which allowed productivity of crops and agriculture to just massively spike, so that we have the potential to feed the population, even potentially up to like 10 billion people. Through this Haber process. The only problem with the Haber process is, again, it requires really high temperatures and very high pressure as well too. Okay, so that's not things that are going to happen in living organisms. However, there are some organisms that can do what we call fix nitrogen into useful forms. So this fixation allows us to react nitrogen with hydrogen or oxygen to create compounds in here. Many of these are single-celled prokaryotes. They often live in symbiosis with plants. They may live around the root system, so that they're essentially fixing nitrogen into a form that the plant can use to grow. So if you add fertilizer in there, you got to have the prokaryotes that will convert, not always, but to have it in there. Some of them live in symbiosis with animals. If you think of termites, they eat the wood in your house, right? Well, the only reason they can do that is because in the gut of the termite are spirochetes, which can not only dissolve those cellulose bonds, but they can also fix nitrogen into useful forms as well too. There are enzymes in these organisms that can overcome that high activation energy. Okay.
So, what does it mean by fix again?
So fixing is basically reacting inert nitrogen with either hydrogen or oxygen to make a more useful compound.
Oh,
Okay. And we are going to look at these in a little bit of detail here. So that industrial process is, a major step forward. Um, Mimicking biological nitrogen fixation could yield a lot of energy savings as well too, because making that fertilizer, 100 million tons annually, takes a lot of energy. Okay, because you got to have those high temperatures and high pressures. So if we can find a way to do what these archaebacteria do, then we could fix the nitrogen much more cheaply. Okay, so when we talk about fixation, we're really looking at the oxidation states of our nitrogen compounds here. Okay, so we can make our nitrates, our nitrites. Um, those are those titles are wrong. So clearly, this is the nitrate column, this is the nitrite column, and this should be the nitrogen column, and this is correct in the ammonia column in there. Huh, I never noticed that before. But you can see that we are looking at a change in the oxidation state here. Our nitrogen can be +5 or +3 or -3. But when that nitrogen is in an ionic form, it's useful to us because that makes it much more reactive. When it's in this covalent triple bond form in nitrogen by itself, that's very, very challenging to react. Okay, so um, and to be more specific about your question about fixation, nitrogen fixation is going to reduce nitrogen gas to ammonia or ammonium. Okay, nitrification is going to basically add oxygen to our nitrogen to make either nitrite or nitrate. Okay, once we have it in those forms, we can assimilate it into the plants and other microorganisms. So they can reduce nitrates and nitrites um, via various reductases that are in there. They can incorporate ammonia directly into amino acids or other compounds. Once an organism dies, it'll return the ammonia to the soil. So that's why you think about mulching and all that stuff, fertilizing your lawn and whatnot. You're basically throwing dead organic material on there to help things grow. Or if you're into serial killers, there's some guy in, was it Toronto, who was starting a landscape business, and he cut a deal with some woman that she would let him store his equipment in her garage, and and he would keep her yard well for free, something like that, while he was building up his landscaping. Except he was a serial killer who preyed on men in downtown Toronto or something like that. So there were like 7, 8, or 9 bodies buried in various planters on her property that she didn't know about. So yeah, but it's just returning the nitrogen to the ground, right? Dead organic material. Okay. And then denitrification is, we have a nitrate that gets reduced back to nitrogen gas under anaerobic conditions. Okay. So what happens there is our nitrate becomes the ultimate electron acceptor instead of oxygen. That's why it's got to be anaerobic. Okay. So we can look at this nitrogen cycle. And again, I want you to just be able to kind of follow along with this, is that, if we start here at the very top, we have atmospheric nitrogen gas. Okay. Various bacteria and archaea can fix that to make ammonia. Okay. Once it's in the ammonia form, plants can use reductases to oxidize it. Um, so, sorry, to change the oxidation state into nitrites and nitrates, which can be more easily incorporated into various components. And then, of course, we can see denitrification, where we can again make nitrogen gas. Okay. So now we have this little thing in the middle here, okay, which we'll get to in a minute, this anammox reaction. there are some weird bacteria out there. We'll come back to that in a minute. But the main thing is the outer circle in there, where we can take fixed nitrogen into ammonia, use reductases to convert that into nitrites and nitrates, and then denitrify it to get back to the nitrogen gas. Okay, so there are two important enzymes in our nitrogen assimilation: nitrate reductase, which will reduce NO3 to NO2, and nitrite reductase, which will reduce NO2 to ammonia. Okay, that's all I want to say about those. Those require various things, but again, these are typically in plants, and I'm trying not to talk about plants. Our nitrogen fixation is carried out by the nitrogenase complex in here. So again, we're essentially doing the same thing as the Haber process, but we're doing it at physiological conditions instead of at super high pressure and temperature in here. Nitrogenase complex uses ATP, and it moves these electrons around. So you end up, basically to make 2 molecules of ammonia, you use about 16 ATP molecules. So yeah, well, ADP molecules technically. All right. All right, so that's all I want to say about that. We can look at the various cofactors in here. We have all sorts of regions very similar to a lot of GTP binding proteins. There's this really kind of neat FeMo cofactor. So it's got an iron molybdenum in there. So it uses basically molybdenum instead of magnesium. But we're not going to go into any great detail about that. I just want you to be aware of it. Maybe I would ask a question for a take-home question on this, but I doubt it, because again, this is mainly in plants, but it's just another kind of neat, massive complex that's necessary for the start of this nitrogen incorporation. Okay. All right, and you can see here, here's this huge pathway of electrons flowing through all this different stuff. Again, I'm primarily concerned in this course about once we have ammonia nitrate or nitrite. Okay. Alrighty. And of course, it helps that the mechanism of our dinitrogenase is not well understood. But anyway.
So we need to know, like, the Haber process is one way to do it, and then another in the physiological form. Mm-hmm. This is how you do it.
Yes. Yeah. Yep. So living organisms can't exist at those high temperatures and pressures. Except for maybe some weird ones, but we don't know any of those yet. So we have this physiological process that results from a massive series of electron transfers. It takes a lot of energy, but you can fix it to get the same thing. And then once it's fixed into the ammonia form, you can use these various reductases to make your nitrites and nitrates, which are very reactive. Okay. We're not going to sweat this. More than you need to know. Again, more than you need to know in here, but it is kind of neat. If you look at plant at the roots, you will see these bacteria that live along the roots that actually are fixing the nitrogen into a form. So we see this kind of symbiosis that occurs. You really can't grow plants in a sterile environment. They need bacteria. Okay. So let's look at assimilation versus fixation. Fixation is specifically nitrogen to ammonia, and assimilation is nitrate or nitrite to ammonia. So it's 2 different routes to get to ammonia in here. Both of these are electron transfer. Both of them use molybdenum, and they have just a ton of redox cofactors. Our iron sulfur centers, our NADHs and ADPHs, ferridox and flavodox, and so on and so forth. All sorts of those in there. Okay. All right. But our big goal is to get to ammonia or nitrate or nitrite, depending on what we're doing. Okay. So that would be all well and good, except there's a really crazy shortcut. This is, well, I like this, but this is probably less important. You will not encounter this very often in here. But this is relatively new. I mean, I think we're talking late 90s, early 2000s that this was discovered. Actually, it might have been even mid 2000s. I'll have to go back and look at the exact date on that. But I know when I started biochem, we didn't study this because I remember coming across it when I was prepping to teach a lecture for biochem. I was like, what is this? Because it's new, really new. There are some weird bacteria out here. And the Animox reaction is peculiar to some really peculiar bacteria. It short circuits the nitrogen cycle so that there's no denitrification step. It's really kind of neat because it can be used in waste treatment to remove ammonia. And what it does is it essentially has this hydrazine hydrolase that produces this hydrazine molecule, right? And then that hydrazine will then allow you to denitrify the nitrite in there, okay? Um, and produce ammonia. What is hydrazine? It's rocket fuel. Okay, it is. It's stunningly toxic to most living organisms, but not these really weird bacteria. Okay, so this hydrazine can be found in this phylum Planktomycetes. Okay, so toxic reactive hydrazine, very nonpolar, and it diffuses across membranes, so it can break across the membranes. So what we see in this particular Planktomycetes bacteria is that their phospholipids are not really they're made of laterranes instead of our typical ones. So laterranes have these cyclobutene rings that stack. So you can see here a membrane bilayer is much, much thicker. And this prevents the hydrazine from crossing the membrane so easily and helps allow these weird bacteria to do the thing that they need to do. Okay. All right, so it's just a weird little aside in there, but there are shortcuts around this biologically. If I were a little more hippie spiritual, I'd say life finds a way. So like, how can you live in rocket fuel? Well, you got to have laterranes as your phospholipids and hydrazine hydrolase and whatever. All right. So all that is our precursor. The whole big thing here is, hey, how do we get nitrogen out of the atmosphere and into my body? It's like, what was that Austin Powers movie, Goldfinger, and who was this guy? It's like, get in my belly, I ate a baby. It's like, how do I get the nitrogen into me? All right. So once we actually have nitrogen in a usable form, just like we saw with our urea cycle, where glutamate and glutamine are key, and glutamine is kind of like a short-term storage of excess nitrogen. Well, we can use it to transport nitrogen around and use it to get our ammonia molecules to wherever they need to go, so that we can synthesize other things from it. So how do we make glutamine? Well, we can take glutamate, and then we can phosphorylate it to activate it, and then we can remove that lovely inorganic phosphate, which is a great leaving group, and add on our ammonia there to give us our glutamine in there. Okay, and that's good. I would say you're coming in at the right time because the first 25 slides are just like extra stuff, but I'm recording everything, so it'll be on there. Okay, so glutamine synthetase, big enzyme. What does it do? All right, all sorts of stuff in there. Okay, so we can adenylate our glutamine synthetase as well too, which will basically help inhibit it. So if we adenylate it, it doesn't actually inhibit it on its own, but it increases the sensitivity to other inhibitors. Oh my. So you guys ever look at like old time radio or do like ham radio stuff where you have to like tweak the dial ever so slightly and adjust everything, get all lined up so you get a signal in there? I mean, it's like driving through the backcountry listening to AM radio. It's like no one does that anymore. But that's essentially what we're doing with a lot of this regulation here. We're like, okay, I'm not going to pick up the radio here, but I'm going to take my course control and move that into the right area, so it's a lot easier for my fine control to get us there. Our adenylation doesn't actually inhibit glutamine synthetase, but it makes it easier for it to be inhibited. That's the sound of the auto sensor clicking on for a second there too, because it's like, oh, I got a great analogy, but it's awful. And I'm like, yeah, tenure or no tenure, maybe I should not say that, but yeah, all right. Anyway, there are a whole variety of compounds that are involved in this regulation cascade in here. Okay, so we've got inorganic phosphate. What I like is this regulatory protein P2. So you basically have this uridylated P2 that will bind to our what is this, adenyl transferase here, and go through this whole process. And you can basically see that, oof, man, this is a pain. Okay, so our adenyl transferase in there can, let's see, is subject to regular. Okay, trying to figure out how the best way to say this. But anyway, so our glutamine, obviously we can have feedback inhibition. Okay, our uridyl transferase can be inhibited from that. Our uridyl transferase will uridylate P2, which can interact with our adenyl transferase, and the P2, the uridylated P2 can bind to the adenyl transferase, which can play a role in the de-adenylation of our glutamine synthetase in there. Okay, so if that is adenylated, it's more easy for this to bind. Okay, to de-adenylate it and make it active. Okay, P2 by itself can bind to enhance this and to inactivate this. I feel like I'm really messing that up.
You said glutamate that makes it easier for it to happen, but doesn't actually make it happen.
The adenylation of glutamine synthetase. So when you actually add that adenylate group to the glutamine synthetase, it makes it easier for this other stuff to bind to inhibit it. Yeah, I think that's what I'm trying to say. Whew. All right. Although my picture doesn't quite look right here. Cysteine inhibition increasing. No wonder you guys are confused. I'm talking circles around myself. I'm going to blame the 80-degree temperatures in here. All right, but the big key is we have a ton of regulation just stacked on top of regulation, stacked on top of regulation. So P2 regulates the adenyl transferase, which helps inhibit the glutamine synthetase. But if you uridylate P2, then that will de-adenylate glutamine synthetase and make it active. Okay.
If you adenylate P2, is that what you said?
Uridylate P2.
Oh, okay.
Okay. So basically, when you attach a uridine to P2, it makes it more likely that it's going to de-adenylate our synthetase and make it more active. But when P2 is by itself not uridylated, then it makes it more likely that you will adenylate our transferase and inhibit its activity.
What is the AT?
Oh, the DAT, or the AT by itself, just the AT?
Yeah, the AT that's getting attached to the
Okay.
P2 at the top.
That is adenylyl transferase.
Okay.
So it's the one that's going to add the adenyl group to the glutamine synthetase. And it's a reversible enzyme as well too. That's why you see it at both the top and the bottom. So when P2 by itself is bound to adenylyl transferase, it enhances the adenylation of glutamine synthetase. When P2 becomes uridylated while bound to adenylyl transferase, it catalyzes the de-adenylation of that. All right, working our way through it. Okay, so all this is part of the regulation of our transamination reaction that we have got going on here. Okay, and again, like we saw in previous chapters, this is going to involve pyridoxal phosphate, or PLP. Those are an important cofactor for pretty much all of our transaminases in here. And it can form this lovely little shift base with lysine. Lysines in our aminotransferases to help catalyze the reactions. Okay? So here we have a proposed mechanism. At this point, I'm probably not going to ask you guys to know the mechanism. Um, it would be enough to say that, hey, we have this nice little channel in here. Here we have our amino group that's going to get kicked off. Okay. Um, what we can see is we have a lovely little acceptor in here that you know, we can see our cysteine can interact with our omega carbon on the side chain and form this bond in here. Okay, that will release ammonia. Okay, and then the ammonia can then bind to our compound to be whatever our compound is here, one of our groups to form the basically to have the transfer of the amino group. Okay. So all this leads us into amino acid synthesis overview. So hopefully you remember seeing in previous chapters in the citric acid cycle, you could take amino acids and degrade them and get intermediates of the citric acid cycle, urea cycle, so on and so forth, pyruvate, convert them into pyruvate. You had your ketogenic and your glucogenic amino acids, all right? A lot of these are reversible, okay? So we can see here, bacteria can synthesize all 20 amino acids that we incorporate in the proteins. We can't do that. I think we can do 11. Some of them are conditional. It just depends on what we need at the time. But we do need to consume some amino acids as well too. So all amino acids derive from one of 7 different precursors. Oh my. So in the citric acid cycle, we got alpha ketoglutarate and oxaloacetate, OAA. In the glycolytic pathway, you've got pyruvate, 3-phosphoglycerate, and phosphoenolpyruvate. Okay, so those are 3 from there. And then in the pentose phosphate pathway, you've got ribose 5-phosphate and erythrose 4-phosphate. Okay, so those are our 7 precursors. Probably a good idea to know those precursors. Which ones come from where? All right, well, it's just kind of the opposite of what we saw before, but alpha ketoglutarate can be used to make glutamate, glutamine, proline, and arginine. Pyruvate can make alanine, valine, leucine, isoleucine. Okay, that's not too bad. 3-phosphoglycerate, serine, glycine, cysteine. All right, a little weird there. Our phosphoenolpyruvate and erythrose 4-phosphate can make tryptophan, phenylalanine, and tyrosine. So both of those. So we really only have 6 columns in here. Oxaloacetate can make aspartate, asparagine, methionine, threonine, and lysine. And ribose 5-phosphate makes histidine. So that's the only one by itself there. I always found it kind of interesting that you have different precursors for serine and threonine, because to me, they're really similar. It's just that extra carbon group in there is probably why you need 2 different compounds in there. Okay, so because of the way I'm going to do the next exam, I want to go a little quicker than normal. All right. Like I say, next exam is going to be fully take home. Oh, yeah. Sweet. Right. Okay. So how do we get these? All right. Again, we're going to be looking at multi-step reactions here. So glutamate can be derived from the transamination of alpha ketoglutarate, just like we saw back in chapter 18. Okay. Once we make glutamate, we can use glutamate as the precursor for glutamine with our aminotransferase, or we can do some more complex chemistry and get proline or arginine from it. Okay. So how do we get proline and arginine? All right, so we can phosphorylate our glutamate. So again, we're activating our glutamate. And then we can get this glutamate semialdehyde, which non-enzymatically will form basically like our ring structure. If you recall, our glucose can have a linear form, it can have a cyclized form. Well, we'll spontaneously cyclize our glutamate semialdehyde in here to make this purine 5-carboxylate PST. Once we have this lovely little ring structure in here, that will be the substrate for purine carbohydrate reductase that'll make proline. Okay. You do not need to memorize any of these in there, but this is just kind of a neat mechanism for it. Proline can also be synthesized from arginine. Okay, so arginine, we can convert that to ornithine. Ornithine, basically in there to our glutamate, and so on and so forth. Okay. Arginine gets synthesized from ornithine in animals. Again, ornithine comes from our urea cycle. Shoot, I forgot. Well, I'll do it on Wednesday. I'll hand back the exams on Wednesday. I've actually got them with me, and we'll go over them. But because ornithine was the answer to one of those questions on the multiple choice portion. So bacteria have a different synthesis pathway. I'm not going to sweat that. We're not going to go into the great details. Although, maybe we could look at the two different pathways in there. And I don't know. I don't want to make you guys do that. That's pretty mean. Serine we get from 3-phosphoglycerate in the glycolytic pathway. So you can see we see, again, a hydroxypyruvate. We actually get our nitrogen from glutamate. So we make alpha ketoglutarate again. So this very reverse reaction provides our amino group to give us this 3-phosphoserine. Once we remove the phosphate group by a phosphatase, we get our serine. And then, of course, we can eliminate our methyl group here and produce water to get glycine as well. So an extra step from serine to glycine. Okay. All right. Yeah. There we go. Okay. Cysteine will also derive from this. And essentially what we're going to be doing is we're going to take that hydroxyl group on the end and replacing it with a sulfhydryl group. Okay. Yeah, we're not going to sweat the details of that. Okay. In mammals, we can look at this biosynthesis of cysteine from homocysteine as well too. All right. So there's our cysteine. Homocysteine looks like this. Serine looks like this. So again, we're just going to kind of, basically combine these two to give us a cystathionine, and then remove our amino group, our excess amino group there, and cleave that to give us alpha ketobutyrate. Woohoo! Whoa, crazy! And cysteine. All right. Oxaloacetate can give us aspartate, and then aspartate can be used to make asparagine, methionine, lysine, threonine, and you can see pyruvate can be used to make alanine, valine, leucine, and isoleucine. Okay. All right. Good to know, right? Okay. When we get to the aromatics, okay, this is where we start to looking at PEP and erythrose 4-phosphate. So here we have phenylalanine, tyrosine, and tryptophan. Okay, and you can also use phenylalanine to make tyrosine. Okay, so there's two different pathways to go through there. Okay, we have this charismate intermediate. Where's our charismate? There you go. Looks something like this. Okay, what I like about this figure, and if I were to come up with a take-home question on here, we could look at, well, where do these come from? Okay, so our phosphoenolpyruvate is providing the orange carbons, and our erythrose 4-phosphate is providing the blue carbons. Okay. All right, so this is where our charismate is. This is a nice little intermediate. Then we can further react our charismate to produce either phenylalanine, tryptophan, or tyrosine. Okay. All right, so histidine, blah, 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 blah. Big, long reaction, right? Look at that. 7 steps just to get to our intermediate, and then we have a bunch more steps to actually get to our histidine. So it's complex and energy intensive. Okay. One thing that I do think is important for you to know is that many of these pathways share this phosphoribosyl 1-pyrophosphate as an intermediate. So PRPP1. And, like I say, so many enzymes or materials have P's or G's in them. We talked about PLP for our pyridoxal phosphate as an important cofactor. Well, here we have PRPP as an intermediate. Okay. The People's Republic of Pyrophosphate. Now, I don't. Anyway. Okay. The important thing is we make this phosphoribosyl pyrophosphate as an intermediate. It is a highly regulated enzyme that will make this intermediate. So the amount of PRPP that we have can help control the rate of production of various amino acids. Okay. All right. So overall, we have all these pathways that are making amino acids. How do we regulate them? All right. We can't just have a one size fits all regulation. So we have a multi-layered approach where we're tweaking a lot of dials. Maybe I should actually talk to my friend who's a DJ. Yeah, because you got all the different levels and stuff in there. You got to adjust all the different components to get the audio just right. Um, I don't think I have quite much of a musician's ear. I remember I was helping a friend do a sound check, and I could just basically tell him if I could hear stuff. I'm like, I can't tell you about the tone or is this the right frequency? And like, what's the frequency, Kenneth? Oh, an obscure reference. Okay. Um, but yeah, there are all sorts of things that interact with each other to regulate these productions. So, yes.
So the PRPP,
Mhm.
where does it come from?
So it's made by this ribose phosphate pyrophosphokinase. So you have this ribosol intermediate, and then it gets phosphorylated on either end here. And,
sorry, I meant to say, where did the ribose phosphate? Is that just a enzyme?
Yes.
Is that an enzyme that is like already available in the body? Or
yes. Yeah. It's so I kind of think of it as like we talk about one of those regulation things. Was it 2,3-BPG is regulated by what 1,3-BPG, whatever that enzyme is in it. So you have a different enzyme that makes the regulatory molecule. So you have an enzyme that makes this PRPP. So it's just basically pumping out intermediates. So
if you had like a deficiency in this enzyme, then you wouldn't be able to make amino acids?
Most likely. Yeah, I'll have to check on that to make sure. I mean, now that's assuming it's like absent or completely unable. If you don't have PRPP, you can't. I suppose you could eat all the amino acids that you needed, but it would most likely be a really severe problem. If you just had something that inhibited it, then you might see a decrease in here. Because keep in mind, I think we're also going to use PRPP for making our nucleic acids as well too. So this is an important enzyme in here. Okay. So when we look at our different amino acids, a lot of them use really similar pathways. It's like, oh, we're going to make aspartate, and then we can go this way or this way or this way or this way. How do we know once we have our aspartate? How do we know if we're going to make asparagine, methionine, lysine, or threonine? It's going to be, well, what do we need? So if we have lots of asparagine and methionine, they'll feed back and inhibit those pathways. If we're lacking lysine, then we'll kind of shunt towards that pathway. All right. We will see also the use of isozymes for particular pathways as well too. There may be isozymes that are preferential for making threonine as opposed to isoleucine or lysine. So let's keep them internally consistent there too. So one enzyme might be more prone to making threonine. Another isoenzyme might be more prone towards making asparagine. Okay. If we look at the feedback in feedback inhibition, I've been talking too much today already. We can see here, once we make our isoleucine, it can feed back to this enzyme, and we'll keep our threonine because it just kind of feeds back. Yeah, says, okay, I've got enough isoleucine. Let's not, waste more energy to make more of it. Keep it as a threonine. Okay. The isozymes are maybe a little bit less intuitive to understand. So again, our aspartate can give us 4 different products, 4 different amino acids in here. So if we use different isozymes, which can bind different effectors, and they have different affinities for these effectors, then we can produce just one of those amino acids as needed, as long as we are making the right regulatory molecules. So if I need more methionine, but I don't need lysine, threonine, or isoleucine, then I will ramp up the isozyme that makes methionine, and I'll leave inhibitors in place for the other 3. So you basically have slightly different versions, different flavors. Okay. Um, I would say it's kind of like Neapolitan ice cream. I don't know, it was always so cool as a kid. You had like strawberry, vanilla, and chocolate all in the same container, right? But it's like, I really don't like the strawberry. And it's like, oh, I'll eat the vanilla and the chocolate or whatever, so that's what we have here. You have like your particular scoop for each of the flavors that are in there. Okay. And you kind of regulate those. Yeah.
How do the isocycles regulate it exactly? They're just like,
So they are a very slightly different structure, and that difference in structure will allow inhibitors or effectors, could be promoters as well too. Now, promoters is a bad term. Inhibitors or positive effectors to bind with different affinities. So maybe our methionine one has a different affinity for something that turns off the others. So methionine doesn't bind this inhibitor, but the other three isozymes do bind it better because of their slight structural difference. Okay. Maybe an analogy would be like, do you ever see those Honda Civics when somebody's like bolted on a wing on the back? Right. Yeah. Okay. Yeah. It's like the cheap secondary car modifications in there. I always laugh because I had a friend I used to play basketball with, and he had a relatively inexpensive car, but he put thousands of dollars of modifications into it. I mean, it looked kind of cool, but at the end of the day, it was still a Honda Civic. You know. But so that's essentially what we're doing here. You put that wing on the back of the car, it does affect the airflow ever so slightly, and you get a slightly different drag coefficient and downforce on the wheels and stuff like that. So you can see some slight performance changes. So that's essentially what we're doing here. We're making these very small, subtle little changes. I always think I don't know how many people are into racing or something like that, but my dad used to race, and then you look at Formula One and like, okay, we're gonna tweak this, we're gonna give this like 3 rotations, so we change the angle of the wing like ever so slightly, and that millimeter of difference is going to add however many pounds of pressure to change the performance of the vehicle. That's essentially what we're doing with our isozymes. We've tweaked the structure just a little bit to make it more or less likely to bind different effectors, so that you can kind of if you produce those effectors, that means even though you have this protein all running, only one isoform is running at higher capacity to make the amino acid that you're lacking. And maybe you're lacking 2 of the 4, so you make sure both of those are active. It gets, like I say, this is hell. Because
is it kind of like, is the isozyme actually have different components in it, or is it just a conformational change?
It is. So the conformational change results from a structural change. So our enzyme is a protein. It's going to have a slightly different protein sequence. So maybe we've got a valine in place of a leucine. So the size difference is just enough that instead of fitting this way, it fits this way. And maybe that opens up a binding clef or makes it more likely. So the isozymes, they're doing the same reaction, they're catalyzing the same reaction. It's just we're making the regulatory parts more or less sensitive due to structural changes that come about from sequence changes. So, and our gene products too. So you would have like several different genes for the different isoforms, and so you could bring maybe do expressional control as well too. Although some of them could be post transcriptional modifications too. But yeah. So let's take a look at some of these aspartate-derived pathways and some of the regulation in here. So you can see you get all these lovely little feedback here. Here, isoleucine feeds back into the threonine section here. And these are like multi-step processes here. So this goes back like multiple steps in here. Our methionine feeds back to homoserine. Same with our threonine in here. But you can see, our isoforms, you've got an A1, A2, and A3 isoform that gets you to this intermediate, which can then be, converted into either lysine, or if you have this other enzyme in here with a B1 and a B2 that can promote either methionine or threonine, then you've got C1 and C2 that'll lead you on to isoleucine. And, C1 is sensitive to isoleucine for inhibition, C2 not so much. Okay. So the whole big point of this is there are all sorts of subtle little tweaks and much, much more complexity than we really talk about. We tend to say, well, here's the pathway, here's how you make, isoleucine starting with aspartate. But really, there's a bazillion little steps in there, and it kind of depends on which isoform of which enzyme is in there. Unless you are studying this specific reaction in great detail, it's not something that we need to know. What you need to be aware of is there are different isoforms of these enzymes that affect the regulation. And all that allows this fine tuning of metabolism to maintain homeostasis. We want to have the correct amount of whatever biomolecules we need at any one time. Like go.
On that graph right there, that essentially means like if there is enough homoserine
Mm-hmm.
laying around, then it's going to more likely produce lysine instead. It's going to inhibit more homoserine being made. Is that what that's saying? And so it's going to go to the first pathway?
Yes, yes. Now, you see that everything that's inhibiting the formation of the homoserine is in that B2 isoform. So that basically means our threonine and our isoleucine are feeding back to inhibit that. And even our threonine feeds back into the aspartate A3 as well too. But B2 seems to be insensitive to that. So it's still going to make, B2 is still going to be making homoserine, but you'll only make methionine through that. If you make enough methionine, it'll just kind of inhibit that as well there too. So you may build up some homoserine, but your aspartate of beta semialdehyde will then all be shunted into lysine until you make enough lysine, which feeds back to stop that. Oh yeah. Good times, right? Good times. Okay. So that's how we make our amino acids. That's great, right? We got a bazillion steps to make amino acids, and you can make them from each other, and they can be regulated differentially with different isoforms and different effectors and so on and so forth. But that's not all. Yeah, I feel like I should be doing an infomercial up here. Your amino acids, once you have them, can be used to make other things like porphyrin rings for heme or phosphocreatine or glutathione or various neurotransmitters or cell wall constituents or so on and so forth. So let's briefly kind of look at those. If we want to make our porphyrin ring, okay, and recall these porphyrin rings are kind of big conjugated series in here that we'll often put a heme inside of to transport oxygen or carbon dioxide or carbon monoxide or cyanide. Okay, no. But our porphyrin is going to be in the heme, hemoglobin, all that stuff in there as well. The pathway gives us this important intermediate, this delta amino levulinate. Okay. We also have another intermediate that's fairly important, this porphobilinogen. All right. We are going to talk briefly about porphyrin diseases coming up in a little bit. So we're focused on these. So we have this multi-step pathway. We start with maybe glycine, we go blah, 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 blah. Oh, here's our intermediate, delta amino levinylate. All right. We make some more stuff and make some more stuff. And, we get to, sorry, or we can start from glutamate and do this in a few more steps. Okay. Once we get to all that good stuff in there, all right, we can take this amino levinylate and combine this with our porphobilinogen and start building our heme structure in here. Okay. And do we have multiples of these? Like 8, yes, 8 amino levinylates and 4 porphobilinogens can give us this nice little ring structure. We can close the ring structure here. And then once we start incorporating things in here and adding stuff to, adding various tails to make our different hemes and whatnot, modifying in there. But our amino levinylate and our porphobilinogen are our 2 important ones that are building the basics of that. After that, it's just modifying our overall structure. Okay. The important thing is we want to have these ring structures here with our nitrogens kind of pointed towards the center, so those nitrogens can coordinate our heme molecule. Okay. So most animals synthesize their own heme. If you end up with mutations or poor regulation of the enzymes in this passageway, you can get a class of diseases called porphyrias, right? And what this basically means is if something's not being utilized appropriately, you get precursors that accumulate in the red blood cells, the body fluids, and liver. So, for example, if you accumulate uroporphyrin 1, you get very dark urine. So you can, pee pink or pee purple, okay, depending on light, heat exposure, peeing under a UV light. It's kind of neat because in your bones and your teeth, they might glow red under UV light. So, if you go to some cool party where they have black lights and your teeth are red, you might want to get checked out for porphyrin disease. Um, Skin tends to be sensitive to UV light, so it can get damaged very easily, and you get a craving for heme. So, hey, wait a minute, red teeth, sensitive to sunlight, craving heme or blood. Oh my God, you're a vampire. So, yeah. All right. Heme is also the source of bile pigments. We look at bilirubin in here. So heme oxygenase linearizes heme to give you biliverdin, which is kind of green. So if you bump your arm or something like that and get a bruise, that greenish compound in there that shows up is the blood that is being oxygenated to biliverdin, and then the biliverdin gets converted into bilirubin, which is kind of yellowish. So that's why you start off with this big purple bruise with the blood, turns green as you're converting it into biliverdin, turns yellow as it gets into the bilirubin, and then kind of goes away as you heal up in here. So a major pigment of urine, the urobilin, kind of gives urine its yellowish color. And we can further degrade this into like something called stercobilin. All right. So if we look at bilirubin, this is a liver function in here. You can go through this whole fun little pathway here, but this is necessary for dealing with excess heme or old heme, getting rid of it. If you can't break it down, can't break down bilirubin, you end up with jaundice. Newborn babies, to test their liver function, they check bilirubin levels. Until the liver kind of comes online, because that can be kind of one of the last organs to really start working well. You can put the kids under a UV light to help break down bilirubin. Okay. But you'll see this in also people who have impaired liver function as well too, or if they have like a bile secretion issue, maybe gallstones or pancreatic cancer, yay! Or if they lack this transferase to process it as well too. Okay. But UV light will cause that photochemical breakdown to make up for this transferase. Just as a side note, if you're not feeling well, don't read medical stories about like people with weird tumors, or I don't know if you, I didn't see the game, but there was that referee in the NCAA tournament who collapsed during the one of the Gonzaga games, and they discovered he had some blood clot because of the way he came back, and so it was basically collapsing in there, saved his life because otherwise probably would end up having a stroke and dying. But yeah, so I'm like, I've probably got a blood clot, I'm gonna die. No, I'm just tired. Yeah. Anyway, we can also look at glycine and arginine as being precursors of creatine and phosphocreatine. If you recall, creatine and phosphocreatine are very high energy compounds. They're often used for like building muscle. You'll see like weightlifters will make their creatine shakes or whatever. So they got to get huge. But phosphocreatine gets hydrolyzed for energy in the muscle. You basically kind of can make phosphocreatine by combining glycine and arginine, and then using Adomet, right, as a methyl donor. And remember, Adomet is a single carbon transfer molecule, and it's really fast and efficient. All right, and very specific. All right. So if we're going to make phosphocreatine, we need a glycine, arginine, and Adomet. We can phosphorylate it by ATP to be stored as an energy source as needed. Glutathione is typically present in relatively large amounts in the cells. It is a reducing agent and functions as an antioxidant. So it helps basically keep our proteins and metal cations reduced so that we can keep our redox enzymes in a reduced state so we don't have to worry about oxidation and radical formation. Basically, it can help scavenge and prevent oxidative damage. Okay. We can oxidize glutathione to a dimer using a disulfide bond. So we have our GSH, we oxidize it, and you make this GSSG bond in there. And then it can transfer those electrons and become reduced again. Okay. Our D amino acids, and everybody did actually, I think everybody did a really good job on answering this question on the previous exam here, but we can actually occasionally see D amino acids in some weird bacteria. So we normally say D amino acids are not incorporated into proteins. That's true for everything that's not a weird bacteria, but some bacteria can do that, and they can racemize these D amino acids. If you eat a bunch of D amino acids, say if you eat bacterial stew made from these weird bacteria, you'll either directly metabolize the D amino acids or you'll racemize them into the L form, and then you can incorporate them again. Okay. It's really kind of neat. If you get a bacterial infection from one of these, you can use this L-fluoroalanine as a general antibiotic because it'll get incorporated in there. It'll knock down the racemase and prevent the bacteria from making the amino acid that it needs, and kill the bacteria that way. Okay, if we look at aromatic amino acids, okay, they are often precursors to hormones, lignans, and other natural products in there, various compounds. I don't know if you have like vanilla and cinnamon candles or whatever. It's I'm a big hater of scented candles because it's essentially indoor air pollution. It's better to just run stuff through a filter. But anyway, like our alkaloids and these various spices and scents are often compounds that incorporate aromatic molecules in there. Okay. We're not going to talk about auxin. All right. Neurotransmitters, though, are an important derivative of amino acids. So things like our epinephrine or norepinephrine or dopamine or our histamines or our serotonin and our GABA, gamma aminobutyrate. All these are lovely little neurotransmitters in here. If you're like me, go outside and histamines go crazy because of the spring allergy season. We went up into the mountains this weekend for a day, and I was like, oh, hey, this is great. Except I had a really weird headache because of high elevation and the storm that came in. Let me tell you, Boone looks really different when the visibility is 300 feet versus 3 miles. That storm system that came through, I was like, hey, look, kids, we're in a cloud. Can't see a thing. All right. Other important stuff. Arginine is a precursor for nitric oxide. All right. So nitric oxide plays an important role in blood pressure regulation, blood clotting, and so on. It's kind of neat because how does nitric oxide get where it needs to go? Well, it's a gas. It just diffuses. So you have these localized nitric oxide production molecules, and then the nitric oxide just kind of diffuses out there, which is kind of neat for blood flow and all sorts of stuff in there. But you can make this from arginine, get this hydroxyarginine where you've now oxygenated the amino group on there, and then you can produce
I have a question.
citrulline nitric oxide. Yeah.
So that's like that happens naturally in the body.
Mm-hmm.
But like, for instance, I have bad circulation problems.
Mm-hmm.
So I've been taking L-arginine and L-citrulline to try to produce nitric acid to try to help.
Yes, yeah.
Is that a really like something that you can take in a supplement and make happen, or is it really only come from what's going on in your body?
Yes. How's that for an answer? So the theory is if you have excess arginine, it's going to be metabolized. Okay? You don't want to have, your body's going to work towards homeostasis. So all the different ways that arginine can be incorporated, if you're consuming excess arginine, it's going to probably stimulate all those activities. Since one of those is this formation of hydroxyarginine and citrulline, then the theory is you will make more nitric oxide, and that will help with the circulation blood pressure regulation. Possibly with the citrulline, Yeah, I don't know. I don't know. That seems like that would be antithetical, but yeah. And now this is where I wish I'd kept in contact with one of my professors from grad school because he studied L-arginine, and that was his answer for everything. So just keep taking L-arginine and you'll live forever. But yeah, it's theoretically it should, but it would depend. Like if for example, this particular enzyme that's in here, if your nitric oxide synthase is at low levels or something like that, then you're just going to be shunting that arginine someplace else. So it's probably not going to be harmful, but it might not be doing as much as it can. So it would depend on the enzyme levels as well too. And unfortunately, you can't take a pill to make more enzymes. Or do CRISPR mod. Okay, no, never mind. We're getting into the realm where we may be able to start doing stuff like that, stimulating the body. Yes, exactly. Yeah, different ways in there. But theoretically, arginine would push through this pathway to produce more nitric oxide. All right. And then, of course, I guess part of this might depend on your calcium levels as well, too, because you got to have your cofactors in there, and so you got to look at all the components. So I don't know. It's like to dig it out. I don't know if we'll have time. I really want to do it, but there's the why is milk often vitamin D added to milk? Because of the whole calcium and the whole uptake, and there's a cooperative effect in there too. I don't know if your arginine, if you take your arginine with milk or something. I have to look into that. That'll be interesting. I did. That reminds me. I know you'd asked the question a week or so ago about why does exercise stimulate HDL? It's a good question. It seems to, but I have not been able to find any mechanism for why that's true. So what I did find was there were several papers that said HDL levels increase after about 9 months. So you have to, it's like you can change the LDL levels after 2 to 3 months of exercise, but to change the HDL levels, it takes about 9 months of exercise. There are other papers that say in a shorter term, you can change the sensitivity of HDL to cholesterol, but there's not really a good understanding of why exercise may increase those levels overall. So at least not that I could find. So I was like, went down the rabbit hole one day instead of grading labs. All right, where are we here? We only have a few minutes left. Okay, so this is probably a good spot to pause. And on Wednesday, we will finish up this chapter, and we will, I'll remember to give back the exams on Wednesday, and we'll go over the exams. All right, I'm feeling the pressure because I'm like, oh my God, we've only got 5 weeks and we've got 7 chapters. So that's why we're kind of skimming over. These are long, like everything is interconnected in here. Once we get through this exam 3 material, it gets a little better. We still have long chapters, but they're not metabolism, so they're a little quicker to get into the gray matter.
We don't have a final, right?
No, we just have the last exam. Yeah. Okay, how do I do this? Come on. What's that? Probably not. It'll probably be maybe the following Monday. I don't think we'll get there fast enough. Yeah. Stop the recording as well.